为主要的一个资料。
哦。
stop 一下行吗？加那个啊？
哦，可以吧？We will wait for like 2 more minutes for more people to come in. Okay, I think we can get started. Hi, everybody. My name is Maisie. I'm one of the student volunteer of DASHU. First, I want to welcome everyone for joining today's virtual journal club. This is the first journal club of DASHU in 2020. A warm welcome to everyone. And today we are really honored to have Dr. Yuan Ji as our speaker today to introduce a novel rule-based dose-finding trial called i3+3. Before I hand it over to him, I would like to spend 2 more minutes to introduce DASHU for those of you who are new to this meeting. So DASHU is a nonprofit organization founded 5 years ago, and our goal is to provide a virtual community and platform for people who are interested in data science to exchange ideas and share opportunities. Joining us is free, so definitely check out our social media accounts to become a member. And the recordings and slides from previously virtual journal club, as well as today's lecture video will be uploaded at our YouTube channel. So please feel free to check it out and subscribe to our YouTube channel. In addition to our routine monthly journal club, we also host a series of on-site activities and events. For example, in a few months in May, we will co-sponsoring this symposium on role of statistics in cancer immunology. This will take place at the Genentech headquarters in San Francisco. We would like to encourage everybody to register and join this event. We have some old friends of DASHU, for example, Dr. Yuan from MD Anderson, Dr. Liu from Dana-Farber, and Dr. Huang from Pfizer as the keynote speakers. So for those of you who are interested in innovative statistical methods in immunotherapy, please stay tuned. Also, we have another great event in May, which is the 4th Statful Onc Annual Symposium. This symposium is a great opportunity for oncologists, statisticians, and regulatory agents to share ideas and solutions about oncology clinical trials. And we would like to encourage everybody to register for this event as well. Like I've mentioned, DASHU is a non-profit organizations, so donations are welcome, and all the donations will be served to serve the community, since our administrative cost is very low. Last but not least, we need more volunteers, and we need volunteers to help us organize and promote these great events through social medias. As you can see here are pictures of our amazing team members and volunteers, and our team is dedicated to promote data science. And please feel free to reach us. And here is a WeChat QR code on the bottom of the page. So if you're interested in joining our team and meet this great, amazing people, please feel free to scan the code. Next, I want to give you a heads up of our speaker next month. We're fortunate to have Dr. Bob Batista. He is a CEO and co-founder of Dr. Evidence. Dr. Evidence is a technology and consulting company that provides automated research to stakeholders of healthcare ecosystems. So for those of you who are interested in AI in drug discovery, please stay tuned. With that being said, let me introduce today's speaker, Dr. Yuan Ji. Actually, I am a graduate student working with Dr. Yuan. Dr. Ji here at the University of Chicago, and he is an amazing statistician and a mentor, and I'm really proud to be his student. It's my pleasure to introduce him today. So Dr. Ji got his PhD from the University of Wisconsin-Madison, and he is an expert in innovative designs for clinical trials and novel statistical methods for high-dimensional informatics data. For those of you who've worked on phase 1 dose-finding trials, you'll probably know him for his Bayesian designs such as MTPI and MTPI-2. He is also the co-founder of Laya Consulting Inc. Company. This company focuses on innovative and adaptive designs for clinical trials in the drug development. So without further ado, let me hand it over to Dr. Ji. And for the audience, please send your questions through the chat box, and there will be a Q&A session at the end of his talk to answer all your questions. Okay.
I want to thank you for DASHU to invite me for this presentation, and thanks Maisie for the quick introduction. The last item here is my conflict of interest disclaimer. I'm going to go through today a new design called i3+3 design for phase 1 clinical trial. It's a rule-based design and based on a review of the past 30 years of research in phase 1 designs. I have quite a few slides, and so I will probably speak very fast in order to cover them. If you have any questions, please, you can type them currently in the chat box under the Zoom video. And then at the end, if we have time, I'll hopefully get to some of it at least. So this is a joint work with Maisie, who just introduced me here, and Sujian Wang from FDA. So phase 1 trials, I assume many people are quite familiar with. Essentially, it deals with the first human clinical trials for a new drug, and the goal is to find the MTD. So essentially, in a phase 1 trial, we want to set up a pre-specified sequence of, let's say, capital D doses. They're ordered from low to high, and then we climb up and down based on a statistical design adaptively based on the outcome. And outcome here is called the toxicity. It's the type of toxicity that's pretty severe, such as organ failure. It's called dose-limiting toxicity. From statistical point of view, we treat patients at every single dose, let's say dose I. let's say we treat N sub I patients on that dose and then observe YI patients experiencing DLT. And this becomes a binomial sampling, which is very familiar to most statisticians. The probability of the toxicity is usually unknown, denoted as P sub I. And there are a couple of assumptions that we normally impose on any dose finding trials or designs. The first one is quite familiar, especially for cytotoxic agents. We assume that P sub I is less than or equal to P sub I plus 1, which means as we increase the dose, the toxicity is monotone. It also goes up, or at least it's not decreasing. And there's also assumption that we normally impose, which is efficacy monotonicity. And this assumption sometimes gets lost because we get very familiar with the traditional setting of the dose finding. But we normally also implicitly assume that efficacy also increases with dose level. Otherwise, it doesn't make sense to escalate a dose when it is safe. The reason is not to induce more toxicity. The reason for the escalation is to induce more efficacy. So at the end, we want to find the MTD, but the definition of the MTD, at least for many rule-based designs, haven't been very clear. So we insist that one should scientifically define what MTD is. In this case, it can be defined as the highest dose with a toxicity rate that's lower close to a target rate. So we specified, for example, 0.3. So I just want to very quickly go through a hypothetical trial in a graphical format. Suppose in a trial there are 4 doses, and we treat patients in cohorts. And normally in these newer designs, we would insist on specifying this target toxicity rate, PT, the quantity I was mentioning in the last slides. And then we treat 3 patients usually at the lowest dose. Here, the 3 patients are in green dots, which means they don't have any DLTs. Without any statistical modeling, we would want to, for example, make a decision on what the next cohort of patients should be treated. In this case, we want to escalate the dose. And the reason is 0 out of 3 is pretty low and less than our target rate. And therefore, we want to escalate. And keep in mind here, the reason we want to escalate is because we assume the next higher dose is more efficacious, not necessarily more toxic, because we want to treat patients for efficacy, right? So based on that logic, the next cohort of 3 patients are treated at dose level 2. None of them have DLTs. Based on the same logic, we want to escalate to dose level 3. And 3 more patients are treated, and of these 3, 1 of them have a DLT, and the other 2 have no DLTs. And we then compare the empirical rate, which is 1/3, to our target rate. Seems it's pretty close. However, from statistical point of view, 3 is a very small number. There's a big variability associated with that, and therefore we want to confirm if this is really 1 out of 3 or something else. So the decision is to stay and treat another 3 patients. After that, we would look at the total of these 6 patients, and turns out only 1 of them have DLT. We again compare to 0.3. Seems the rate is lower than our target. We want to escalate again. And the trial goes on like this until at the end, 6 more patients are treated at dose level 4, and 2 of them have DLTs. And then at this point, since the target rate of 1/3, this is the empirical rate, 1/3 is pretty similar to our target rate, but with 6 patients. So the question is, should we stop the trial? If we do, which dose should be the MTD? Which one should be the recommended phase 2 dose? Should we pick dose 3 or dose 4 as the MTD? These are the questions that we're faced with. And more importantly, should we actually really believe that there are, with 6 patients on each one of these 2 doses, that we're how confident are we about the MTD? So these become statistical inference question, and traditionally we stop at this point and then normally pick dose level 3 as the MTD. Although if we truly believe the target of 0.3, then there's no problem if we want to select dose level 4 as the MTD as well. There are very other difficult cases we start to see, especially in the current immune oncology era. There is sometimes we see no DLTs at all. We keep escalating until the highest dose, and then nobody has DLT. Then the problem becomes, for example, what should be the MTD? As I was mentioning at this point, perhaps we should look at efficacy. If the dose is safe, this is a good problem. It means that the drug is pretty safe, but the question is it efficacious? So that's the question that needs to be addressed. And sometimes the MTD is sandwiched between two doses, in this case, dose level 2 and dose level 3. And perhaps in this, the right decision when this happens is to insert a new dose. And we do that quite often in clinic setting. And also sometimes the designs are just not good. For example, we see very safe outcomes in the first 3 doses here. And then on dose level 4, we get a 2 out of 3 DLTs, but some designs would still want to escalate because of the borrow information. There's nothing wrong from the statistical point of view. You want to borrow across all the 4 doses, but the escalation to dose level 5 can be very risky, and from practical point of view, because we're treating humans in these clinical trials, such decisions may not be accepted by review committees. So in this case, it's a bad design in practical sense. So the 3+3 design is probably one of the most well-known designs for phase 1 trials. And this is a chart we published a few years ago to describe the simple algorithms of 3+3. The key here is that there's no statistical models and inference. Everything is based on fixed rules and essentially logic. This design has been dominant for the past at least 3 decades in the clinical setting. But it seems like there's a movement that there are more and more needs and desire to use non-3+3 designs, more innovative designs. So what the main problems of the 3+3 designs are, first, that it's not flexible. It doesn't give rules when you treat not 3 or 6 patients at a single dose. It only tells you what to do when 3 or 6 patients are treated. For example, what if you have 2 or 4 or 5 patients? And you may wonder, how can you get that? If the cohort size is 3, how can you get 2 patients or 5 patients, for example? It's because you might get dropouts from the trial. And when that happens, usually based on 3+3, you need to backfill another patient, but that takes more resources and time. The main problem statistically for 3+3 is the immature stopping. Essentially terminates a trial pretty randomly and treats no more than 6 patients at any given dose. As statisticians, we know that 6 is not such a big number. It's not a magic number either. For phase 2 or phase 3 trials, we never just treat six patients and say, oh, we know what the drug, how the drug performs. But in phase 1, somehow we stop when there are 6 patients treated based on 3+3. So we should be able to go beyond 6 with novel designs. So these are just 2 examples recently to show that the 3+3 is not as efficient or safe even as other adaptive designs. So to start talking about other new designs, the key question actually here is to define the MTD properly. If you look at the 3+3 design, it never defines what the target is, what the MTD should be. It's essentially based on a rule which says you don't want to treat, you don't want to select the dose with more than 2 out of 6 DLTs at any given dose. But scientifically, we want to define what MTD really is. So we are advocating the following framework. First, we want to define this target toxicity rate as the MTD rate. With the clinicians, you can elicit such number. You can by asking the question, for example, at MTD, if we treat 100 patients, what would be the highest number X that would allow to have DLTs? And that X gives us a elicitation of this PT number. However, again, as a statistician, we know that we normally don't hit exact number. Let's say the X is 30, then our PT is 30%. But we don't really believe that in a phase 1 trial, we would have a dose that with a probability of toxicity exactly equal to 30%. So we would want to allow some wiggle room or some variabilities associated with that target. So we can continue and ask the physician about the following questions. For example, what about 29 patients? Would that still be okay, acceptable as an MTD? How about 28? How about 31? So by going up and down, you'll probably reach a boundary, and that boundary gives you an interval, which is called an equivalence interval. The equivalence interval is intervals that you elicit from physicians that covers the target. So not only we know what target we are aiming for the MTD, but with the equivalence interval, we have a wiggle room to allow doses that fall into that interval to be also considered as the MTD. So the claim is that if we are able to decide the two values, the PT and the equivalence interval, then there's a bunch of designs that can really work very well. So this is a very quick review of what the innovative trial designs for phase 1 trials in the past 3 decades. At the bottom here is a 3+3 design. At the top here is what we're going to introduce, the i3+3 design. But through the past 3 decades, there are different branches of model-based designs. Some people would call some designs model-assisted. I don't like that name. I would still call it model-based because either you use a model, you don't use a model. For the some designs use complex models such as CRM and BOIN. Some designs use simplified model like independent beta binomial models, which are seen in some of these designs. And then today we're gonna go back and say maybe we don't need a model back 30 years later after the 3+3 paper was published. And this, I call this a spiral evolution. We went from rule-based design to model-based design back to rule-based design, but with smarter rules. So why do we want to go back to rule-based designs? At this point, you may have several questions in mind. For example, didn't we just mention that rule-based design like 3+3 is not good? And then why should we trust this new rule-based design called i3+3? What's the difference between this new design and others? How does it perform? And this is the questions we want to address for the rest of the talk. But to start, we first want to look at a set of new design called the interval designs. You probably have heard about some of these names like TPI, MTPI BOIN design, or MTPI2, which is equivalent to the keyboard design. So as I promised earlier, if you are willing to specify the PT, the target, and the equivalence interval, then all of these designs, the interval designs, can help you to design a phase 1 trial. So all of them share the same operation tool, which is the decision table. So if you specify a target and the equivalence interval, all these designs will give you decision table somewhat look like this. They might be presented in different formats, but essentially they tell you exactly what to do during any possible step of the trial based on any possible outcomes. And the columns here are the number of patients treated at any given dose. The rows here is the number of patients from those who are treated at the dose that have DLTs. So for example, here, if you treat 3 patients at a given dose, then you might have 0 patients, 1 patient, 2 patients, or 3 patients having DLTs. And these rules in terms of escalation, stay, and de-escalate will tell you exactly which dose to use for the next cohort of patients. And this covers all the possible outcomes. Suppose you have a sample size of 21. And the table can, in theory, be made for any sample size. And so with this table like this, then you know exactly what to do for the trial. And the table is automatically generated based on the models or the rules of these interval designs. The tables across all of these designs are, in vast majority of them, have the same identical rules. It's just a little bit different in some observations, which I'll cover a little bit. But this is the commonality that's shared by all these designs called the interval designs. You input the target and the equivalence interval, and the output is a decision table like this. So this is a real case study that's published in 2014 based on one of the interval designs, the MTPI design. And I'm gonna just very quickly show that how this was conducted based on the table I just demonstrated. So this is the real data of the trial. It has initially started the trial at the 90 milligram with 3 patients treated. Nobody has DLTs. Then you go to the column 3, row 0, and read out the decision, which is to escalate. So the decision was to escalate to the next dose level, which is 135 milligram. On dose level 2, the paper reported 6 patients treated, and 1 of them have DLTs. Then you go to column 6, the same table. Go to column 6 and row 1 here, which is again an escalation. So you escalate to the next higher dose. Initially, the trial had the next dose as 200 milligram. So they went to 200 milligram and treated 3 patients, and all 3 of them had DLTs. And then you go to the same table, column 3 at this point, but the row 3, and the readout is D and U. D means de-escalate. U means this dose needs to be eliminated. It's not acceptable anymore because there's too much toxicity. So dose level 4 is eliminated. The trial was de-escalated, but the investigators they were pretty smart. They didn't go back to the original 135 milligram. They inserted a new dose, 150 milligram, and then de-escalate to the middle dose and treated 6 more patients here. And none of them have DLTs. So in theory, you go back to the table, you read out 6 and 0, which is another escalate. But remember, the next dose, higher dose is eliminated, so there's nowhere to escalate to, and the trial stopped and picked the 150 milligram as the MTD. If you want to, you can actually insert another new dose between 150 and 200, because since 150 is also pretty safe. But that was a real case of using the table. I think since around 2015, there's some debate about what table is optimal. For example, this is a table based on the MTPI designs, and there've been criticism about some of the decisions that may not be practically safe. For example, the table, this is a table for a target of 30% and the equivalence interval between 25% and 35%. So that's where the MTD is roughly specified. And then the table says that when we have 4 patients treated and 2 of them have DLTs, the decision is stay. Same decision for 6 patients and 3 DLTs, but not the same for 8 and 4. So in all these 3 cases, the empirical rate is 50%. It's above the equivalence interval or the target rate. But the decision when there are 4 patients and 6 patients are stay. And this prompts some practical concerns in terms of toxicity and risk on the patients. And the argument is that the MTPI may not be as safe and should be modified. However, if you read some later papers, this is caused by something called Occam's razor. It's the way of Bayesian, a Bayesian way of saying model selection, penalizing for complex models. And you can see that the Bayesian models actually can automatically learn that when we have higher sample size at 8, even though the empirical rate is still 50%, at that point, the model recognizes that there's enough evidence to show that this is indeed the 50%, and that the decision becomes de-escalate, which is what we want. But when the sample size on these doses are small, like 4 or 6, even though the empirical rate is only 50%, there's still a large variability associated with that evidence, and therefore the model recommended stay. Nonetheless, in practice, this is perceived as problematic, especially by IRBs, by regulatory bodies, and therefore there is a need to improve or to get rid of these practically unpleasant rules. So this is one of the solutions is called the MTPI2 design. It eliminates or blunts the Occam's razor, which is shown here. essentially, it's not a good thing to do from statistical point of view. It's essentially saying that when you select models, you don't care if the model is more complex or less complex. It's not what we want for in principle from statistical point of view, but we do this nonetheless to satisfy the regulatory and the safety requirement. As a result, you can see there are a lot of state decisions from MTPI is changed to non-state decision like de-escalate or escalate. So this makes the MTPI2 design more nimble. Essentially, it doesn't want to stay at a dose. It actually wants to go up and down more swiftly. So this satisfies the regulatory requirement because, for example, here the state decision and here the state decision are changed to de-escalate. So safety-wise, there's not a concern anymore. But is this a better design? It's definitely more costly because you would change more doses more often in a real trial. But it's nonetheless it's deemed safe and from regulatory perspective. I also want to change direction slightly different to the other two interval designs. One is called a cumulative cohort design, which was published in 2007. And then the other one is the BOIN design, which is extension of the CCD, the cumulative cohort design. Both designs uses a point estimate, which is the empirical rate. It's the same as let's say the 3+3 design. You just look at what happened from the data. It's the yi divided by ni. And then they use intervals as a decision boundary. So essentially, remember we asked you to specify two things: the target probability of MTD and the interval plus and minus this target. That's the equivalence interval. So both designs use interval as a decision boundary instead of calculating the probability of these intervals, which is the inference for the TPI designs. And this is the table that summarizes the decision. So in all three designs, the decisions are stay, escalate, or de-escalate, but the inference is different. For the MTPI design, the decision is based on this posterior probability of the intervals. But for the two CCD design and the BOIN design, the decision is based on directly compare this point estimate or the empirical rate with the interval. So statistically, this is unorthodox because this doesn't involve any variability measurement. You're using point estimate for the inference. Again, we start to see that there are many of these interesting developments that kept simplify statistical principles in order to make the design simpler and more practical. So a little bit of explanations on the BOIN design because I know it's getting quite popular in practice. The BOIN stands for Bayesian Optimal Interval Design. So the first question is it Bayesian? Because if the inference is based on this point estimate, then it seems like it doesn't have to be Bayesian. The paper was written treating this as a posterior mode using a beta binomial distribution, but it can also be perceived as an empirical rate. You don't even need a model for this. And this is actually the motivation for the i3+3 design. Another thing is it optimized because the O stands for optimal interval design. The optimization based on the original paper was based on an ad hoc criteria. As an end result, what happened is that the boundary in the BOIN design is changed. Essentially, two additional quantities were added to the equivalence intervals as the new equivalence interval. And this was considered as optimal interval. But the intervals actually, in my mind, it cannot be optimized. These are user-provided quantities. It's something you want to define for your MTD. So you can optimize that. It's for example, elicit from the discussion with clinicians. But to summarize, these are the two types of interval designs. The first one is based on the posterior probability of these intervals, and the other one is just using interval as boundaries, using the point estimate or the empirical rate of the DLTs outcomes as the inference quantity. So one more thing about both the two types of design. I was saying that these are equivalence intervals. So PT is the target. These are the two equivalence interval boundaries. They are nominated based on elicitation from clinicians. They are also used as the decision boundary in the actual design. This is the case for both CCD and MTPI. In the BOIN case, there was an optimization procedure that changed the original nominal boundary into the decision boundary. And the decision boundary actually is nested, is smaller than the original boundary, which left a gap. This is the two quantities I was talking about, the E1 and E2. And these E1 and E2, these two quantities are not a function of sample size. They actually do not diminish when the sample size goes to infinity. So asymptotically, this has a problem. The problem is that if you have a dose actually fall into the gap, based on the nominal boundaries, that dose can be considered as the MTD. But based on the decision boundary, the dose is outside the equivalence interval and will not be considered as MTD. And that's not asymptotically diminishing, meaning this conclusion will stay there no matter how big of the sample size is. And this to me has some problem theoretically. However, this boundary is usually very small. And then with small sample sized phase 1 trials, we usually never have a dose that fall into the boundary, or the boundary or this gap cannot be realistically estimated. So the empirical performance of BOIN designs are pretty good, are very good actually. So how do we compare different designs? Other than the theoretical properties, what we care is the frequency operating characteristics. So we run simulations based on truths, which generate data from these true probability of the toxicities, and then we repeat this multiple simulated trials, applying each one of the designs, and we look at the summary statistics, for example, the probability of finding the true MTDs, the patient's allocations where they are treated, and we also look at the toxicity profile of each design and compare the sample size. So this is one way of comparing designs. There are other ways of comparing designs. With these newer and better interval designs, we can actually directly look at the decisions that we would make. For example, this is a decision table based on one of the interval designs, and we can present this to the physicians, to the safety monitoring committees prior to the trial is started and get agreement. So we can, this is very useful in practice. But for other designs like CRM designs, although we cannot get a table like this prior to the trial, we can actually run simulations and then summarize the operating characteristics in terms of the frequency of decisions under the CRM design, for example. So in this case, you would actually see that, for example, here, when 3 patients are treated and one of them have DLTs, sometimes the CRM would escalate. Most of the time it would stay, and sometimes it would de-escalate. So tables like this to summarize frequencies can also be used to compare designs. And the tables like this are actually quite informative to clinicians so that they understand that the CRM decisions sometimes is not obviously logic from the clinical standpoint of view. And then the question is there an optimal design? Does one design outperform any other designs in all scenarios? The answer is typically no, because each design has some assumptions in their modeling, their rules. So whenever the assumption is satisfied, that design becomes the optimal design for that assumption. So when we simulate based on different scenarios, sometimes the scenario with assumption agree with the assumption of one design, sometimes the scenario agree with another design. Therefore, there will never be a design that outperforms all other designs in all scenarios. So go back to the main topic of today, which is the i3+3 design. It sounds like there are so many good designs out there that one can choose. So why bother with a new design? And especially why bother with a model-free and rule-based design, which for the past 30 years, the statisticians have been saying it's not good. Well, let's first have a very quick recast of what happened in the past 30 years. So 3+3 design was first proposed as a rule-based design. It uses basically the x divided by n, which is the empirical rate for inference. Then the CRM design was invented as the first model-based design, has a statistical superiority based on principled statistical inference, borrowing information across doses. But in practice, it needs some overdose control. It needs safety rules to curb the escalation when it's skipping doses, and then it leads to aggressive decisions. So therefore, CRM was kind of modified and reined in using rules like escalation with overdose control. In other words, borrow information in principle from statistics works pretty well, and also the parametric modeling, but it's truncated based on these ethical constraints. And therefore, the model-based idea is simplified in these interval designs. They don't use parametric curve to model the dose response curves. Instead, they use independent beta binomial distributions, but use up and down decision rules based on the intervals, right? And then later, it's even more simplified. Instead of calculating probability of intervals, which accommodate variabilities, the newer designs essentially use the point estimate. If the point estimate is all we need, then the question becomes, can we do it? Can we design the trial actually without a model if we just need a point estimate? And the answer seems to be yes. So this is what we proposed in the i3+3 design. It's an algorithm essentially summarized in this table. Again, we ask users to provide two input. One is the target, the other one is the equivalence interval. And then the design is summarized in 5 decision rules. Suppose at the current dose D, we have enrolled N patients and X patients of them have toxicity outcome. Then what you do is look at the empirical rate, X divided by N. You ask if this rate is below the equivalence interval, meaning if it's less than 0.025. If that's the case, then we escalate. We treat the next patients, the cohort of patients at dose level D+1. That's an escalation. If this point is inside the interval, then we continue at the same dose level to treat the next cohort patient. If this rate is above the equivalence interval, then what we do is we ask one more question, and this is what so-called smart rules come in. We ask to calculate the x-1 divided by n. Take one toxicity out of the data and calculate the new rate, and ask if this rate is, where is this rate? So if the observed rate is above the equivalence interval, and if this lower rate is actually below the equivalence interval, then what this is saying that with 1 unit of DLT, the event taken out from the data, we can jump from above the equivalence interval to below the equivalence interval. How can this be possible? It usually is possible when n, the sample size on the dose, is small, and therefore the one change of the event is influential. In other words, the sample size at the current dose is so small that we cannot even detect or have meaningful inference on the equivalence interval based on 1 unit change of information. In that case, then we shouldn't believe that this empirical rate is confidently above the equivalence interval. And therefore, the decision is to continue. We want to accumulate more evidence until we make sure this is indeed above the equivalence interval, something else. So this is the only exception when the empirical rate is above the equivalence interval. Otherwise, if it's above the equivalence interval and if one change of the unit event leads to inside the interval, then means this is indeed also above the interval. It's pretty confident, then we should de-escalate because we exceeded the range. If both are above the interval, then we should definitely de-escalate. So these are the 5 rules. And then our claim is that these 5 rules will give you a performance as good as any of the previously mentioned designs. So these are the 2 quick examples to demonstrate what we mean by using these 5 rules. So suppose we currently have 3 patients and the target is 17%. Equivalence interval is from 12% to 22%. When we have 3 patients treated, we could have 0 DLTs, in that case, the decision based on i3+3 is to escalate. But when we have three patients treated, one of them have DLTs, then you can see that here the empirical rate is 1/3, which is above the interval. But the one minus that decision, the rate is 0. Therefore, this falls into this line here. The empirical rate is above, the next lower rate is below, then the decision is instead of de-escalate, but stay. And this is actually what 3+3 does. So even though the empirical rate is above the target and the equivalence interval will still stay. And for the other two cases, they are all de-escalate. And for 6, when the sample size is larger, then we see that it's essentially based on the empirical rate and where it's located relative to the intervals. So, and before we run any simulations, we can easily compare the new design with other interval designs by just looking at the decision tables. So this is a comparison of 3 designs, i3+3, MTPI, and BOIN. For 2 cases, when we enroll 6 patients or we enroll 3 patients, you can see largely the decisions agree across all 3 designs. There's exception in either one of these 2 cases. The first one is when 6 patients are treated and 3 have DLTs, then the i3+3 decision is to de-escalate, to go down. And the MTPI is to stay. And that was the criticism for the stay decision. And the BOIN design would also de-escalate. So since i3+3 and BOIN, in this case, are conducting the clinically acceptable decisions to protect the risk. And then in the other case where there are 3 patients treated and 1 of them have DLTs, in this case, actually i3+3 design and MTPI take the stay decision. They do not want to change the dose level. In this case, the target is 17%, and the equivalence interval is from 12% to 22%. And in this case, actually, BOIN design is more nimble. It actually wants to go down. Okay, so you can see that it seems like in both cases, the i3+3 design is doing what clinical or clinicians want to do in practical trials. And it's based on these five rules. So again, as I was saying, that nowadays we don't have to run massive simulations to know if a new design performs well or not. We can just look at these decision tables. You can see that by comparing i3+3 with MTPI and MTPI2, most of the time the decisions are identical. There are only a few cases where they differ. Then you know that the simulation results won't be that different. Like all the other designs here, there are 2 safety rules. One is early stopping. You stop the trial if the first dose is too toxic, or at the end you select the MTD based on this isotonic transformation as a statistical inference problem. And that the performance can be summarized again based on simulations. And as expected, the i3+3 performs quite well. Here, the 0 bar here is the benchmark, which is i3+3. We benchmark all other designs against i3+3. So 0 means i3+3. Above means the design is safer in terms of toxicity rate. Below it means it's not as safe. And you can see here, BLRM is very safe and 3+3 is very unsafe. And the other designs like BOIN, CRM, MTPI, MTPI2 are pretty similar to the i3+3 in terms of safety. And similarly for reliability, the middle designs here that were developed based on models and intervals are pretty similar to the i3+3 design, but the BLRM and 3+3 are the 2 outliers. And then we are not surprised again because the decision rules are very similar across these designs. We've done other sensitivity analysis to see the performance of the i3+3 design based on different width of the equivalence interval. We want to change the equivalence to be narrow or wider. And turns out what happens is when the equivalence interval is wider, the performance in terms of finding the MTD and then safety actually gets better. The reason is that when we enlarge the equivalence interval, we're essentially loosening our criteria of MTD. we are allowing more doses to be qualified as MTD, and therefore the trial becomes easier, and therefore the performance of the designs gets better. So in general, what you see is that with the same target, as the equivalence interval becomes wider and wider, all these performance metric goes up, which means the design performs better and better. We also try to look at the sensitivity in terms of cohort size. One thing I want to mention, I forgot to mention, is that the rules here has nothing to do with the cohort size. Even though the name of the design is called i3+3, you don't have to treat 3 patients at a time. Essentially, it's actually A plus B design. You can treat any number of patients and follow these 5 rules. So we did a sensitivity analysis using different cohort size. We treat 2 patients at a time, 3 patients at a time, 4 or 5, or random. And just want to see how the design performs. And turns out they're quite similar in terms of the safety, toxicity, and reliability. The little bit outlier is the cohort size 4, where the performance is a little better than the other cohort size. But that's unfair comparison, because our sample size was 40. When we use cohort size 4, we usually end up with a sample size of 32 in our simulation, because 32 is divisible by 4, but 30 is not. So in general, the conclusion is that the cohort size doesn't seem to change the performance of the i3+3 designs. Currently, there are free software and commercial software, and this is my conflict of interest from either website. The top one here is the RShinyWeb. The lower one is the commercial website. There are already applications based on these designs that pass the regulatory reviews in practical trials using the i3+3 design. And apparently the one advantage of this i3+3 design is that you can easily explain the design to physicians. These are the 5 rules. You just need to tell them what the 5 rules are, and then usually physicians understand why you're doing this. So it's completely transparent, just like a 3+3 design is. And so that's the advantage of this design when compared to the others. So this almost concludes the presentation. In other words, based on this new research, it starts to show perhaps for phase 1 trials, especially in oncology, a rule-based design with smarter rules might be sufficient. So in other words, model-based inference plus ethical constraints in practice, they keep knocking off our statistical principles that we normally would apply to other inference problems, but not to phase 1 trial designs. So at the end, it's almost equivalent to having a smart algorithm. And the physicians usually are in full control, because they're the ones who actually prescribe the drug and the dose. So they need to agree to the decision. And that's why it's important to have a transparent design. And seems that the i3+3 design is a good one, even though it's not model-based. I have a few slides to show that we should go beyond the existing regime, and then the field is already doing that. For example, the dose is safe, then let's escalate. I kept mentioning that the reason we do that is because we assume the drug is cytotoxic. The higher the dose, the more efficacy. But there's some new development in immune oncology where that assumption is wrong. The higher dose may not lead to higher efficacy. In this case, then you would want to consider other types of designs, like you want to consider both efficacy and toxicity as joint outcomes in the dose finding. Maybe use biomarkers, use other type of design tools to accommodate this new development. And also, some of the folks in the industry knows that this revolutionary drug pembrolizumab from Merck, it was based on a so-called 1,000 patient phase 1 trial. Initially, it was a naive 3+3 design for a dose finding study for the drug. And then at the end, the phase 1 protocol includes almost more than 1,000 patients with sub-studies. But I don't think it's fair to call this a phase 1 trial. It's a master protocol or platform trial incorporating multiple sub-trials. And that's what it is. But this was allowed back in 2014. and led to the approval of the drug. So the MTD should never be the goal for drug development. It's just a first step to establish the safety profiles. And there are other things we should consider. For example, we should consider seamless designs rather than just have a very clear boundary of I'm going to do dose escalation and then consider the rest of them the goals next. The FDA now has the new draft guidance allowing dose expansion cohorts right after the dose escalation. When the monotonicity assumption breaks, then we can consider efficacy and toxicity as joint outcomes. And also, there are more and more combinations like pembrolizumab or PD-1 inhibitor based on another type of drug, like a dual agent combinations. Now we are also seeing three drug combinations, so on and so forth. So I'll stop here and then to see if there are any questions, I'd be happy to address. Thank you.
Thank you. Thank you, Dr. Ji, for such an enlightening presentation. I think we can continue with the Q&A. And the audience, please feel free to leave your question in the chat box.
Okay. So there is a question from the audience. The question says, are there any guidance on how to recommend size for next cohort? I assume this is based on the a design with a decision table. So the question is, when we actually run a trial, let's say we run 3 patients at the dose, is there guidance on how to what cohort size is for the next set of patients? Normally in protocol, one would specify the cohort size like 3. So you would if you follow the protocol, then you would treat 3 patients as the cohort size, or as always. But in practice, different things could happen. For example, sometimes one would actually have a dropout from the 3 patients, and then they would only have information about 2 patients. And suppose if the decision is to escalate, then there's a choice of enrolling 3 more patients or 4 more patients, depends on where you are. Sorry, when we have actually 2 patients and one of them has DLTs, then the decision is to stay, then you would need to enroll more patients. And the question becomes, do we want to enroll 3 patients, which would go to 5, or 4 patients, which would go to 6? Either one is fine, because the table actually tells you what to do for either cases. But the advantage of going to 6 patients is that you would actually end up having a little bit more information to see if this is indeed a good dose, and then allow you to go back to multiples of 3 for the future cohorts. The next question is, what would the comparison of BRM versus i3+3? So we actually showed this plot. So BRM is a very interesting design. Again, like CRM is based on very sound statistical principle. It uses logistic regression as the dose response curve, and actually the inference is based on intervals. However, it needs to have escalation with overdose control, EWOC. And the default value for that EWOC threshold is 0.25. If you use the default value, and you'll probably have experienced this same phenomena as what we have seen, is that the BRM becomes very safe, like very conservative. It doesn't escalate as quick. So which shows here. So 0 is i3+3. This plot shows that the BRM is above 0, which means it doesn't induce as many DLTs as i3+3. It's doing very well in terms of safety. But the trade-off is the next plot. This is the reliability, it's the probability of finding the true MTD at the end of the trial. Because it's very safe, it doesn't like to go up, escalate. And when the MTD is at a higher dose, it's having difficulty finding the MTD. And therefore you see here, the bar is below 0. 0 again is the benchmark i3+3, and the BRM is below that. So this is consistent in what we observe that if we use the default threshold for EWalk, then we see a very conservative BRM. But one can change that threshold, but we're not interested in, for example, how to optimize BRM. Rather, we want to see based on the default value how other designs perform against the i3+3. Okay. There are two people asking a similar question. The question is, could you tell us more detail of a real world example of a drug or trial that leveraged i3+3 design that performed more robustly? Is there any phase 1 study using i3+ approved by FDA recently? The answer to both questions is yes. Let me see. Let me go to, sorry. There are multiple cases where the IND filing have been approved. I wouldn't call this approval of the design because FDA doesn't use that language, but we didn't encounter many comments when we submitted this. But there's no trial have been finished based on the design because the design was only published last year. There are some trials going, but we haven't seen real data out of the i3+3 design yet. I think the robustness of the i3+3 design is highlighted in these two tables. Is that in both cases the decision picked by i3+3 is different from other existing designs, but seems to agree more with clinicians. So that's the advantage. When we use this design and discuss this with clinicians, they usually like what's presented here. For example, they don't want to de-escalate when 1 out of 3 patients have DLTs, and this is what i3+3 does, but not, let's say, MTPI2 or BOIN design. Another question is, what would be the difference in ending results in choosing the target of 0.2 versus 0.3? So the question is, what would be the difference if we use a target of 0.2 or 0.3? The difference is in the decision table. So the decision table based on the either of these interval designs, including the i3+3 design, will change if you use 30% as the target or 20% as the target. If you use 20% as the target, then there will be more de-escalate and fewer escalate because we're targeting a lower rate. We would not allow more escalation if we use 20%. And you can imagine that, therefore, the trial will be more conservative because of those decisions.
And the last question, yes, the video as well as the slides will be uploaded to YouTube. So feel free to check it out. Yep.
Okay.
I think that's all the questions and time's up. So I want to thank you again, Dr. Ji, for joining us today. And I want to thank you all the audience for coming, listen to our talk. And the videos will be uploaded to the YouTube account. So please check it out and subscribe to our YouTube channel. And that's the end of the talk. Thank you.